Officially, good morning, everybody. Hi, good morning on this July Monday morning. We have a very special seminar today. We have our great friends who came to Olsztyn all the way down from Taiwan, from 8,500 kilometers away from here. We are very happy to host you here in our institute, in our almost brand new infrastructure that you always have a chance to see after the open seminar. So today our distinguished expert is Professor Andrew Chi-Wei Huang from Guang University. He's a leading expert in behavioral neuroscience, investigating the mechanism underlying psychiatric disorders and drug addiction. And he will tell us all about it. And we have this pleasure to host you here, thanks to our great friends from the University of Warmia and Mazur. Thank you so much for giving us opportunity. Great applause. To have you.
Thank you.
And I'm giving the floor to Professor and enjoy the lecture.
Okay, good morning, ladies and gentlemen. My name is Andrew Huang from Taiwan Guang University, from the Guang University in Taiwan. And today I will talk about my studies and experiments, especially for the drug addiction. The opioid use disorder. Okay, so today you can see my title is Paradoxical Effects of Opioids in Opioid Use Disorder. Okay, after the DSM-5, if you are interested in the psychiatrist disorder, this disease, the opioid use disorder, we call that OUD. Before that, we call it opioid addiction, because we want to prevent the bias terminology, addiction. So currently we use the opioid use disorder, okay? Okay, it's very far from here, you know. A long and excellent connection since 2016 with Ania, Professor Korzaska, as my girlfriend, and we have 10 years for cooperation, and we take the airplane for 24 hours, you know, So it's very long distance and very great connection. And here is our university, the front door. You can see it's very beautiful place. And here is the campus view. Here is the university car and the sports activity place. It's a very beautiful place. Okay, right now I would like to introduce the outline. The first one, I want to ask what is the opioid use disorder, and the reward systems in the brain. After that, I would introduce our hypothesis, the paradoxical effects hypothesis of abuse of drugs. Okay. After that, I would ask 7 questions, and to develop the hypothesis. Okay. Besides the section 5, you can see I will introduce the novel ideas for treatments. Okay. Finally, is the final conclusion, and the final section is the other hypothesis for drug addiction. Okay, what is opioid use disorder? It's a very severe psychiatric disease in the world, and create a long, it's like this one. It produces a very higher the prevalence rate in the world. For example, the United States and the Iraq, Iran, and Afghanistan. Okay, almost 26.8 million people suffer from this disease, opioid use disorders in the world. And you can see it creates a long-term the social problems in the world, and it can separate the 2 different phases. Okay, the first one is the intoxication. This one, opioid intoxication, it's under the acute treatment of opioids. Okay, on the other hand, you can see the second phase is opioid withdrawal phase. Okay, and after the termination, stop using the drugs, the abuser will produce the withdrawal symptoms. Okay. However, in my lab, we focus on the intoxication phase. Okay, and we found that this addiction can produce not only a rewarding effect, but also the aversive effect. Okay, and this kind of aversion, the effect is not this kind of aversion effect. This aversion effect is a withdrawal symptoms. Okay, so in my study, we emphasized the intoxication phase, and we found this drug, the opioid, can produce simultaneously the reward and the aversion. Okay, such like euphoria for the reward, and dysphoria, agitation, empathy, judgment impairments for the aversion. Okay, so you can see over here, the other labs, they examine the withdrawal symptoms. It is another kind of aversion. Okay, after abstinence, okay, stop using the drugs, and the abuser can produce the opioid withdrawal signs or symptoms. It's another kind of aversion. Besides, the abuser produce the comorbidity, the disease, such as depression or anxiety disorders. And besides, most of them, the abuser produce the suicide attitudes. And finally, they produce the complete suicide, okay, to die. Okay, these slides you can see our textbook teaches the reward. The brain, the abuser just enjoy the reward. Okay, the textbook told us when the abuser use the abuse drugs can produce the reward effects, and the reward system in the brain you can see over here. This brain area from the ventral tegmental area (VTA) project to the nucleus accumbens. Okay, this pathway we also call that's a kind of mesolimbic dopamine pathway. Okay, that's the textbook told us the abuser enjoy the euphoria. Okay, underline this pathway. However, in my study, we found that not only the reward, we call that a paradoxical effect. Okay, since 2008. In my lab, we collect a lot of the data, and we found that abuse drugs such as morphine, amphetamine, and alcohol can simultaneously produce the paradoxical effects, reward and aversion in the brain. Okay, so we call this kind of the hypothesis is a paradoxical effects hypothesis. Okay, and today I just focus and present the morphine. Okay, this drug's addiction. Okay, because we want to test the behavior. One is aversion, and the other is a reward. So we use the two different behavior tasks. One is aversion task. We call that the condition test aversion. Okay, you can see over here, the animal, rat or mice, drink the sweet solution such as saccharin or the sucrose. And after that, the rat or animal was given the morphine and produced the condition test aversion like this one. The animal reduce the consumption for the saccharin solution. Okay, so we call that a kind of condition test aversion, and especially to test the aversive effects for the drug. Okay. On the other hand, we also use another kind of the behavior task. It's a reward task. Okay, it called that condition preference. Okay, during the pre-test, we put the rat or mouse in the compartment of the CPP, the condition preference, the box. Okay, and then we put the rat or mouse in one compartment of the CPP box. Okay, it was given the morphine. Okay, on the other hand, the rat put on the other date in the compartment of the CPP box. Okay, was given the saline, and finally post the test, the rest was put into the CPP box. We measure and test the span time for this compartment and this compartment. Okay. Here is the results. You can see the morphine groups the rats spend a lot of time to state the paired side of the CPP box. Okay, and here is the setting. Okay, so it was produced the reward effect. Okay. So we use this kind of aversive task and this kind of rewarding task. Okay, after that we ask 7 issues. Okay, the first one, what kind of we want to ask can morphing simultaneously induce the rewarding effect in CPP task, the condition preference, and the aversive effect in condition test aversion? Okay, in my behavior data, we show that the y-axis is intake volume of saccharin. Okay, you can see more than the 10 milligram per kilogram the morphine, the dose can produce very severe the condition test aversion. Okay, decrease the consumption of the saccharin solution. Okay, over here, and at the same time, we also to test the condition preference. You can see more than or equal to the 20 milligram per kilogram the morphine can produce the very higher the span time for the parasite. Okay, so because that's a condition preference, it's kind of a rewarding effect. Okay, so we can see equal to 20 milligram per kilogram or more than 20 milligram per kilogram the dose can produce at the same time produce the condition preference. So this is our summary. You can see here is a aversion and here is a reward, and more than or equal to the 20 milligram per kilogram the doses can produce the morphine, can produce the paradoxical effect, okay, the aversion and the reward. However, the 10 milligram per kilogram of the morphine dose just only produce aversion, okay? Okay, besides, we also to test the neural substracts, okay, not only the behavior. Based on the previous literature, we choose some candidate brain areas, like this one. Okay, these 3, it's MPFC, the medial prefrontal cortex, the sub-areas, the CG1, PRL, and IL. And here is central amygdala. Okay, these 3 brain areas, based on the previous literature, they were involved in the aversion, okay, behavior or learning. Okay. On the other hand, you can see the blue color. Okay, these 4 brain areas, that's the hippocampal the sub-areas, the CA1, CA2, CA3, and the dentate gyrus. And the nucleus accumbens and the peripheral cortex. Okay, these 1, 2, 3, 4, 5, 6, these 6 brain areas were involved in the rewarding behavior or learning. Okay, based on the previous literature. Okay, and also you can see these 2 brain areas, lateral hypothalamus and the basolateral amygdala, were involved in the reward and aversive effects. Okay, the behavior. Okay, so we choose these. Many sub-areas of the brain to be our candidate brain areas, and use the C-force. Okay, we use the immunohistochemical staining, this approach to label the C-force and the P-ERK. Okay, these 2 specific proteins to determine which brain was upregulation the C-force overexpressed. Okay, so you can see over here, this is a conditioning phase, and it's the extinction phase, and here is the reinstatement phase. And you can see during the conditioning phase, okay, CG1, IL, and the PRL, and the basolateral amygdala, nucleus accumbens, and the dentate gyrus. Okay, there are 6 brain areas that were involved in the conditioning, and they show that's a higher C-force overexpression. Okay, and extinguish the phase just only 3 brain areas, and during the reinstatement, okay, the recover again the addiction behavior, and it show very higher 1, 2, 3, 4, 5, 6, 7. These 7 the brain areas show the higher the overexpress of the C-force. Okay, over here. And the PRK also show the similar pattern from the conditioning phase, extinction phase, and the reinstatement phase. Okay, you can see the during the resettlement phase, and the 1, 2, 3, 4, 5, 6, 7, the brain areas was shown that overexpressed per p-ERK. Okay. So from this study, we get a summary. Okay, in behavior, the data equal to or more than 20 milligram per kilogram of morphine can simultaneously produce aversion in CTA, condition test aversion task, and the rewarding effect in the CPP, condition price preference effect. So our results is not similar to the previously the textbook, you know, not just only the reward. Okay, at the same time we also tested aversion. Okay, this kind of effect, and you can see the IHC data. Okay, you can see the C-force and the p-ERK, the protein overexpressions in the rewarding and the aversive brain areas. For example, you can see the IL, infralimbic cortex, prelimbic cortex, basolateral amygdala, and the nucleus accumbens were expressed in the C-force and the p-ERK during conditioning. However, during extinction, almost all the brain areas were extinguished. Okay, after that, on reinstatement, the C-force overexpression occurred in the IL, PIL, BLA, CEA, nucleus accumbens, and the lateral hypothalamus. Okay, so in my study, the first study, we get the result. The morphine injection maybe during the conditioning and the resettlement, the phases produced a non-homeostasis and the disturbance of the brain. Okay, so abuser guys, they do not feel good, not just only feel euphoria. They are poor guy because they at the same time feel very bad in the brain. You know, not only the euphoria, at the same time they feel the very bad aversion effect in the brain. So we call that a kind of non-homeostasis disturbance in the brain. Okay. Okay, the second section, the second issue, I want to ask whether the rewarding VTA and the aversive PAG were involved in paradoxical effects. Okay, in this study, we used another kind of approach. We used the behavioral pharmacology, and they used the NMDA. It is kind of neurotoxin to destroy the VTA or PAG. And you can see over here, this one. Here is the NMDA plus morphine, and this one is the PBS plus morphine. Okay, it's a control group over here, and you can see NMDA microinjection to the VTA, and it's destroyed the VTA's neurons. It can attenuate the condition test aversion. Okay, it attenuates the aversive effect. However, you can see the PAG. Also, we use the NMDA plus morphine. This group, there was a non-significant between this and this groups. Okay, so the NMDA microinjection into the PAG did not affect the morphine. Okay, induced the condition test aversion. Okay. However, this one, we test CPP. Okay, it's a reward effect. Okay, you can see the y-axis is a different span time. Okay, in the paired side. Okay, you can see the NMDA, this group. Okay, in the morphine group, decrease the pair side span time. Okay, however, destroy the PAG. There was non-significant between these two groups. Okay, so we get the behavior results. Okay, the NMDA region, the VTA, destroy the VTA display areas, simultaneously reduced the morphine's condition suppression. Okay, it's kind of aversion, and also reduce the reward. Okay, over here. Okay, however, the NMDA microinjection did not affect the PAG. Okay, induce the CTA and the CPP. Okay, so here is our summary. You can see VTA is very important for the paradoxical effect of the hypothesis of the brain areas. Okay, however, the PAG was not important. Okay, besides we asked issue 3, the third question. Okay, does the brain show the neuroinflammatory responses after the morphine induced the CTA, the aversion, and the reward in CPP? Okay, in this study, we label another kind of the protein. We label the neuroinflammatory, the responses. The protein is the interleukin-1 beta. Okay, we want to see the whole brain, which sub-areas was involved in the neuroinflammatory responses. Okay, so here is our data. You can see here is the infralimbic. Okay, it's a sub-areas of medial prefrontal cortex, and the basolateral amygdala. Okay, it's a sub-area of amygdala, and these 4 brain areas was the hippocampus sub-areas. Okay, you can see almost the whole brain produced the neuroinflammatory responses. Okay, and you can see IL, BLA, CA1, CA2, CA3, and DG. These brain areas produce a very upregulation the IL-1 beta overexpression. Okay, however, there was non-significant between the setting and the morphine for CG1 and the PRL. Okay, so we get a summary for the issue 3. Okay, you can see the overexpression for the IL-1 beta it occurred in the IL, BLA, CA1, CA2, CA3, and day gyrus. Okay, so almost the whole brain, such as MPFC, amygdala, and the hippocampus produce the neuroinflammatory responses. So we think that's another kind of aversion, So the poor guy, the abuser, did not only enjoy the euphoria, they also suffer from the neuroinflammatory responses in the brain, So we want to think about how to cure their disease. That's very important. Until now, we do not have a good medicine to treat this disease. Okay. Okay, here is the issue 4. We asked that were the neural activity of MPFC, nucleus accumbens, and basal amygdala affected by different doses of morphine. Okay, in this study, we test a lot of doses of morphine, and we want to see how the sub-areas of the specific brain, their connection, the neural pathway was shifted. You can see that. Sorry, you can see over here. Okay, here is the CTA test, the data. Here is lower dose, the setting. Okay, mid dose, the 20 milligram per kilogram morphine, and 30 milligram per kilogram morphine, and the 40 milligram per kilogram morphine. Okay, here is high dose of morphine from low to high. Okay, doses of morphine, and you can see over here. We use the Pearson correlation to test the C4 data. Okay, to test between the PRL and the nucleus accumbens core, the correlation. We found that it showed that a positive correlation between the PRL and the nucleus accumbens core, and the IL has a positive correlation with the nucleus accumbens core. Okay, you can see that the shift to this one, the PRL and the IL also have the positive correlation with not the nucleus accumbens core, shift to the nucleus accumbens shell. Okay, it's positive correlation. Okay, this one we skip that. You can see CPP, the data the same. Okay, from the 2 milligram per kilogram morphine. Okay, the PRL has a positive correlation with the nucleus accumbens core and the nucleus accumbens shell. Okay, the positive correlation, and the shift to the high dose of the morphine from the IL. Okay, from the PRL shift to the IL, and the correlation has a positive correlation with the nucleus accumbens shell, and has a positive correlation with the basolateral amygdala. So, okay, in this study we get the results. Okay, we extend the paradoxical effect hypothesis of drug addiction in morphine. Okay, we found that the neural association and the neural pathways will contribute under varied doses of morphine. Okay, so we can see here after the CTA test, the crucial connection, it was shifted from the PIL and IL to the nucleus accumbens shell and the core. Okay, and after the CPP test, the connection also shifted from the PIL and IL to the basolateral medulla and the nucleus accumbens. Okay, this study we just want to extend our hypothesis. Okay, so this is our summation. Okay, the MPFC's PIL-IL connect to nucleus accumbens core and shell. Okay, for the aversive effect. Okay, morphing induced the CTA, and the MPFC's PIL-IL connect to the basolateral medulla and the nucleus accumbens. This pathway were involved in the morphing's reward. Okay. Okay. Besides, we asked issue 5. Okay, we want to see the stress, the factor, how the stress to modulate or affect the opioid use disorder. Okay, we call that opioid addiction. Okay, and you can see we use the full shock stress. Okay, in this study, and you can see the behavior data. Okay, here is a CTA. You know, his aversive task, intake volume of saccharin consumption. You can see this group, the full shock plus morphine is one, and this one is morphine over here. Okay. Okay, the full shock attenuate, okay, or block the morphine-induced condition test aversion. Okay, so the stress could attenuate the aversive effect. At the same time, we also to test the reward. Okay, you can see this one. Okay, this one is a morphine, just only morphine injection produced the span time, okay, very higher span time, okay, it's a reward effect in CPP. And this one just only use the full shock also to produce the condition preference, okay. And this group is very interesting. You can see that full shock plus morphine can also produce the condition preference. Okay, we translate the data to this one, and you can see this group, the full shock plus morphine, is very higher the CPP score compared to the control groups. Okay, so when the full shock, okay, it can enhance the rewarding effect. Okay, from this data we can see that. At the same time, we also to test the immunohistochemical staining with the C4, the protein, and we can see the PIL, these brain areas. There was a significant effect for the full shock. Okay, not drug, the morphine, not interaction, and IL also obtained the similar results. However, you can see the basolateral medulla. Okay, the full shock show that significant effects, and the morphine, the drug, show the significant effect, and the interaction between the full shock and the morphine also produced a significant effect over here. Okay, so we get the result, the summary. In our data behavior, you can see the full shock stress attenuated the aversion and facilitated the morphine, induced the rewarding in CPP. So some guys, when they get very huge stress, maybe they would produce the addiction for alcohol or for some abuse drugs. Okay, so it's very, okay, and here you can see the IHC the data. Okay, the C4 expression in the PIL, IL, and the basolateral medulla were higher. Okay, after the full shock affecting the morphine-induced CPP and the CTA. So we can, in this question, we get the summary. The full shock can reduce the morphine aversion and enhance the reward. Okay, and through modulating the neuroactivity of the PIL, IL, and the basolateral medulla. Okay. Okay. The issue 6, we asked another way. We use the environmental enrichment. This manipulation is a kind of anti-stress. Okay, manipulation. The environmental enrichment is is a device. The rats put the cage, and the rats or mice can get a lot of the toys and the running wheel and the partners. Okay, we call that or tuners or shelter. Okay, we call that environmental enrichment. Okay, it can anti the stress. Okay, so in our study, we also see the result. Okay, it unpublished. We just to test the CPP, the condition preference over here, and then we can see the morphine can produce the very higher the rewarding effect in CPP task. Okay, and the EE, the environmental enrichment plus the morphine can reduce the effect. Okay, so it shows that there was non-significant between the unpaired side and the paired side. Okay, over here. Also, we tested the C-force. Okay, after the behavior parts, and you can see here is environmental enrichment and the morphine and the interaction between the environmental enrichment and the morphine. Okay. And we can see over here the environmental enrichment can down-regulation the C-force overexpressions in the CG1, PRL, IL. Okay, these three brain areas are the medial prefrontal cortex. Okay, and then here is the nucleus accumbens. Okay, here's a rewarding the press. Okay, the neural subtracts. Okay, also down-regulate its C-force overexpression, and the central amygdala and the dentate gyrus, okay, also down-regulate its C-force overexpression, okay. And for morphine, this factor just only, you can see over here, the nucleus accumbens upregulation of C-force overexpressions. The others, there was non-significant between the control group and the experimental group. Also, the interaction did not show the effects, okay, over here. Okay, so in summary 6, the question 6, the environmental enrichment can attenuate the morphine, induce the rewarding in CPP, and the CG1, PRL, IL, and the nucleus accumbens, okay, central amygdala and the dentate gyrus were lower in the C-force overexpression. Okay, so here is our results. The EE, environmental enrichment, reduced the morphine's reward in CPP through the CG1, PRL, IL, nucleus accumbens, central amygdala, and then dentate gyrus the neural activities. Okay. In my lab, we also use optogenetic approach. It's a very pioneer approach. Also, in my lab, we use a chemical chemogenetics approach. Okay. However, this time I just present my optogenetic data. Okay, optogenetic simulation data over here. Okay, and this issue 7. We asked that does optogenetic stimulation in the medial prefrontal cortex. Actually, we stimulate its sub areas, CG1 singular cortex 1 and the prelimbic cortex and its infralimbic cortex. Okay, and modulate the stimulus valence. Okay, from reward and aversion to neutral status in the morphine's condition test. Aversion test. Okay, what is optogenetic? Okay, this picture is very good. Okay, the first one to get the genetic construct. Okay, it's a DNA sequence. Okay, and this sequence can produce the light-gated the ion channel. Okay, finally. Okay, and insert in the neural membrane. Okay. And after that, the construct the DNA sequence insert into the virus. Okay, the virus was given into the microinjection into the specific brain areas. Okay, after almost 1 month, 4 weeks, and insert optical fiber into the stem or the target, the brain areas. Okay, after that, turn on the light. Okay, the light can control. It's a kind of light-gated ion channel. It's a new channel, not original the channel. Okay, turn on turn on with the light and turn on the light channel. The sodium ion can go into the membrane. Okay, produce the EPSP excitatory postsynaptic potential. So it's a kind of excitatory stimulation. Okay, so in my study, we use the channel reducing 2. Okay, it's an excitatory stimulation. Okay, you can choose the inhibitory photostimulation. That's okay. Okay. However, in my study, we use this one. Okay, and you can see the figure A. It's a verification for optogenetics. You can see micro-injected the virus into a CG1, and here's the CAMK2. It's a promoter. Okay, and you can see the virus infections in this specific brain area. And also we label the C-force with the red color. Okay, it means this neuron produced the neural activity. Okay, and merge this picture. Okay, and this one also merge these two over here. Merge these two pictures over here, and the right side is optical location. Okay, the optical location over here, and so this picture just a verification for optogenetic approach. And here's our result in behavior. Okay, you can see here's the rewarding valence. Okay, the first this fast we just the rest was consume the saccharin solution. Okay, over here is a kind of a reward effect. So we call it a rewarding valence. Okay, after that compare with the morphine. Okay, produce the condition test aversion. Okay, it's change the valence to the aversive valence. Okay, and after that, we're using the extinction procedure. Okay, the saccharin solution from the aversion to the neutral, the valence for animals. Okay, so these three brain areas, we use the optogenetics channel reducing to excite these brain areas. And you can see the important one is PRL, this brain area. It can modulate the rewarding valence and the aversive valence and the neutral valence. Okay, so the PRL is very important for the modulation, modulate the valence of stimuli. Okay, and the CG1 just only can modulate the rewarding and the aversive. However, it cannot modulate the neutral valence. And IL cannot modulate the reward. However, it can modulate the aversive valence and the neutral valence. Okay. Besides, we also to test the C-force overexpression. Okay, you can see the PRL, this brain areas photostimulation, and during the conditioning, you can see the BLA, VTA, the C-force or down regulation. Okay, down regulations expressed, and during the extinction phase, up regulation the nucleus accumbens core over here, and down regulation the infralimbic cortex and dentate gyrus. Okay, so this data is very important because you can think about this one is PRL. Okay, you modulate these brain areas, and they can affect many the neural pathways. You know, it's a kind of neural networks. So some sub areas down regulation and some brain areas up regulation. So you need to think about its neural networks. Okay, you modulate this brain areas can affect this one, and this one, the brain areas. So we get a summary. PRL, the photostimulation, could modulate the neural activity of the nucleus accumbens, IL, and DG to neutralize the valence of stimulus. Okay, from reward and aversion valence. Okay, maybe you will ask me, how is it important? You can see it is very important over here. It's another kind of novel ideas. Okay, maybe we can think about how to cure these poor guys, abusers. Maybe you can think about use the atypical neurotransmitters such as the BDNF or glutamate neural inflammation or P2X7 the receptor. Think about alternative the method to try to cure the disease, opioid use disorder. However, in my study, we provide altering the variance of stimuli for reward and aversion. Like this one, is a reward effect can to modulate this variance, rewarding variance to the neutral, and also we can modulate the aversive variance of the stimuli to the neutral. Okay, if the stimulus obtain the neutral property, maybe it can cure the disease. Okay. So here's our final the conclusion. First one, the morphine injection during conditioning and the resettlement. It's very important phases and show that non-homeostasis and disturbance in the brain. Not only the euphoria in the brain. Okay, it also produced the aversive effect in the brain. So it showed the non-homeostasis and the disturbance in the brain. And the issue 2, we obtained the results. The VTA is very important because it contributes to the aversion and the reward induced by morphine. And the issue 3, okay, we can see that the morphine can produce the neuroinflammatory responses. Okay, it's another kind of aversion. Okay, and the issue 4, the results show that PIO-IO, based on the different doses of morphine, can shift their pathway. Okay, from the PIO-IO to the nucleus accumbens core and the shell. Okay, and from the PIO-IO connect to the BOA and the nucleus accumbens. Okay, and the 5, we want to see the stress or this factor, how this factor affect the paradoxical effect. Okay, it can reduce aversion and enhance the reward. Okay, by morphine. Okay, through modulating the neural activity of the PIO-IO and the BOA. Okay, and the issue 6, the EE, the environmental enrichment, reduced the morphine's reward. Okay, in CPP through the CEG1, PIO-IO, nucleus accumbens, central amygdala, and the dentate gyrus neural activity. Okay, issue 7, the PIO photostimulation can modulate the neural activity of the nucleus accumbens, IO, and the dentate gyrus to neutralize the valence of the stimulus from the reward or the aversive the valences. Okay. Okay, and this side you can see the other hypothesis for the drug addiction. Okay, the George Koop is a very big guy, in the field. Alcohol addiction or opioid use disorder. Okay, in his hypothesis, he emphasized the 3 stage. Okay, the beginning is the binge intoxication. Okay, is the reward effect and translate to the withdrawal negative effect. Okay, it's another kind of aversion. It's not similar to mine, the reward. Okay, and translate to the preoccupational anticipation. Okay, it's like the relapse behavior or craving behavior. Okay, it's also another kind of a reward. Okay, and translate to the binge again. Okay, intoxication and binge, drink a lot or consume the opioid a lot. Okay, so in his hypothesis emphasized these 3 stages. Okay, however, his aversion emphasized the withdrawal symptoms. Okay, so his aversion is not similar to my hypothesis. Okay, on the other one is Solomon. Is very popular. It's very famous one. Okay, he proposed the opponent process theory. Okay, in the beginning, okay, the first few stimulus of abused drugs and can produce the reward in the brain, and also turn off terminate the drugs and produce the withdrawal the effects. Okay, is also another kind of aversion. Okay, it's not similar to me. It's also similar to the George Koop. Okay, it's the withdrawal the effects. Okay, after many stimulation of abused drugs. Okay, produce a very fewer euphoria, the reward, and produce very higher the withdrawal symptoms. Okay, so in his hypothesis, the Solomon emphasized the opponent process, the reward one and the withdraw another one, okay? Okay, this is my paper, and we published in the current addiction reports last night. This review paper told about this story, the paradoxical effect hypothesis. And I would like to thank my partners, Xu Baichuan and the author Tsai from the Academia Sinica, Taiwan, and Anna Koroska, and my student, pharmacist, Wu Jiwen. And also I would like to thank the National Science and Technology Council of Taiwan. Okay, here's my currently I'm leading this topic issue. Okay, maybe if you have a good article, you can submit to our topic issue. Okay. Thank you for your attention. Thank you. Yeah.